Hello, I'm David Tabb, a professor in the Division of Molecular Biology and Human Genetics at Stellenbosch University. And today I'd like to give you a quick tour of some of the bioinformatics that exist for amplicon sequencing. So let's go ahead straight into our overview. We're going to start with software that's been designed to help us design PCR primers. From there, we'll move on to talk about filtering for base call quality, to be able to decide which sequences we trust, which parts of it we need to truncate. And from there, we'll talk about a very important concept in microbiome sequencing, particularly that of mapping to operational taxonomic units. So we'll talk about some of the toolkits that exist for clustering and for libraries there. And finally, we'll move on to estimating the deleteriousness of non-synonymous single nucleotide variations. And finally, we'll cut into evaluating the quantities of the variants of genomic sequences that we can find based on these amplicon sequences. So how do you decide what is an appropriate primer to use in DNA amplification? We have several different rules that we end up applying, and these represent kind of a best practices rather than a quantifiable set, but let's just try to walk through this. First off, the process of PCR amplification requires that we are able to selectively anneal and release the primers from the sequence by changing the temperature, by thermocycling. As a result, if you are designing pairs of primers that need to work in concert, it doesn't really make sense to employ one that is very GC rich and the other that's very AT rich, because they're going to have different temperatures at which they are released from the hybridization. So we want to design primers that are going to release at equal temperatures. Next, we want to design primer sequences that are highly specific to the target sequence that we're trying to amplify. If you have a very short primer, its ability to be very selective, to only align to DNA next to the gene of interest, is going to be somewhat limited. At the same time, we must create efficient primers. If you think about it, each cycle of PCR, of polymerase chain reaction, has the potential to double the amount of DNA that you have present in your sample, double the target DNA in your sample. If you are able to design efficient primers, you're able to get very close to that theoretical doubling of this amplification process. If you are able to produce inefficient primers, they may release a little less well, or they might anneal less effectively, and that can diminish the efficiency with which you're amplifying. What if you only get 50% more DNA with each cycle, for example? And given that the whole point of the exercise is to polymerize DNA that's complementary to your target strand, it's very important that you not inhibit the DNA polymerase. And it is possible to design a DNA primer that interferes with the polymerase. So that's obviously a no-go. And generally, we just speak of not having any substantial homology among the primer sequences. I realize that that's perhaps a little vague, so I wanted to show a couple examples of this. I've drawn this diagram in this case from a paper from 2012. So A, B, and C here are all trying to show different primer-primer interactions. So when we look at the elements of A, we see some homoprimer interactions where the same forward primer interacts with itself. So you cannot have a sequence within your primer that's complementary to itself. That's going to be very problematic, and you end up with these interactions between different strands of DNA. When we look at the case of D, however, we're still looking at a complementarity, but now it's within a single sequence. We know from the example of tRNA, for example, that nucleotide sequences can be complementary to themselves internally. And so if you can form hairpin loops within your primer sequences, that's not going to work out very well for your reaction. So having these interactions where the primer sequences are interacting with each other, or with themselves, or with other copies of themselves, is basically a no-go zone. When we get down to E, we see that we have interactions between primers and templates. And in this case, we want to be sure that the primers interacting with the forward strand are the only ones interacting with the forward strand, and that the primers interacting with the reverse strand are the only ones interacting with the reverse strand. So that may seem relatively simplistic if you have just a single target that you're trying to amplify in a particular reaction. If, however, you are trying to to amplify multiple targets all at the same time, finding these different sets of primers such that they no longer have the possibility of interacting with each other can become a rather difficult problem. These are all detailed in this 2012 article from Untergasser. Now, there are two really major implementations of primer picking that I want everyone to know about. The first of these is called Oligo7. Now, Oligo7 is, of course, the seventh in a line of tools for this purpose, but it was originally published all the way back in 1989. So you can see that people have been thinking about the problem of optimal primer design for quite a long time. The oligo.net website will give you access to the software and you can download it. And I would note that it's not just limited to PCR amplification. We may not talk all that frequently about silencing RNA, but that's another example of methodologies we can use to try to diminish the expression for a particular gene. So if you're trying to do a PCR experiment, that's great. Oligo7 has you covered. If you're dealing with silencing RNA, you might need to have a toolkit that supports it, like Oligo7 as well. And it's also very good for restriction analysis. Restriction enzymes, of course, are used for chopping at specific palindromic sequences, most typically in DNA. So if you're trying to figure out where, for example, BAMH1 should chop in a given genome, Oligo7 is prepared to field that kind of problem for you. So this is an example of commercial software. There is a free version available, but it's only for walking you through its tutorial, basically. But it can help you to understand what this software is going to be able to accomplish for you. Now, the NCBI also offers a service for primer selection called Primer3Web. Presently, it's at version 4.0, but as you can see, it was originally published back in the year 2000 by Rosen and Skaletsky. The website for the software is right here at bioinfo/EBCEE/mprimer3. And importantly, this is the software that lies behind the Primer Blast interface. So if you're making use of the National Center for Biotechnology information for the website for the service for picking out primers, behind the scenes, Primer3Web is running on your behalf.